Elements of Transferable Antibiotic Drug Resistance, Part 2. Transposable Genetic Elements. Transposons, insertional sequences, mu bacteriophage, and integrons are transposable DNA elements. They can get inserted into the chromosome or plasmids of bacteria. Barbara McClintock got Nobel Prize in 1983 for discovering of these jumping genes. Recombinant A independent recombination system usually occur in transposons in a random fashion between non-homologous sequences of DNA. These play an important role in antibiotic resistance gene transfer between the bacteria. Transposons and insertional sequences, they both are incapable of autonomous self-replication. Both are flanked on each end by inverted repeats, essential for transposition process, and they must exist on a replicon, that is chromosome, bacteriophage or plasmid, to be replicated and maintained in the bacterial population. Transposons differ from insertional sequences in that they encode functional genes that mediate a recognizable phenotypic characteristics, such as antibiotic resistance marker. Transposons associated with antibiotic resistance fall into three major classes based on the general structure and methods of insertion: Class 1, Class 2, and Class 3. In Class 1 and Class 2 transposons, transposition can be either autonomous or non-autonomous. They both consist of composite and non-composite transposons. Composite transposons have two copies of insertional sequences elements on either end. Transposase is encoded by one of the elements. Central region contains antibiotic resistance gene. For example, TN10, that is class 1 transposon. Simple or non-composite transposons do not terminate with insertional elements. They contain short inverted repeats at each end, contain antibiotic resistant genes, and encode their own transposase. For example, TN3, that is class 2 transposons. The third class is the conjugated transposons. They are capable of excision and transfer from the genome of the donor bacteria to the recipient bacterium via conjugation. Their transfer is not constrained to closely related bacteria. For example, TN16 to TN1545 family of transposons, which have been found introduced into 50 different species and 24 different genera of bacteria. Conjugation of the conjugative transposon begins with excision of the transposon from either the bacterial chromosome or plasmid DNA. The transposon DNA circularizes, and conjugated transfer of the single-stranded DNA copy of the recipient proceeds in a manner identical to the plasmid conjugation. A wide variety of antibiotic resistance genes have been discovered on large conjugated transposons in gram-positive bacteria. An insertion sequence is a short DNA sequence that acts as a simple transposable element. They are small, carry the genes necessary for their transposition. Coding regions is usually flanked with inverted repeats, named according to the form ISN, where N is the number. They occur as a parts of a composite transposons, and two insertion sequences flank one or more accessory genes, such as an antibiotic resistance gene. Transposable elements are assigned into two classes according to their mechanism of transposition. Class 1 transposable elements, that copy and paste, and cut and paste in Class 2 transposable elements. Class 1 transposable elements is copy and paste. For example, TN3 and mu bacteriophages, transposition is catalyzed by transposase enzyme. The following image is showing the transposition process. Class 2 transposable elements, cut and paste transposition mechanism does not involve an RNA intermediate. Transposition is catalyzed by transposase enzymes. Some transposases non-specifically bind to any target site in DNA, whereas other binds to the specific DNA sequence targets. Transposase makes a staggered cut at the target site, resulting in the single-stranded 5' or 3' DNA overhangs. This step cuts out the DNA transposons, which is then ligated into a new target site. Process involves activity of the DNA polymerase that fills in gaps and of a DNA ligase that closes the sugar phosphate backbone. DNA integration elements, also known as integrons. Stokes and Hall gave the concept of integrons in 1989. These are sequences of DNA that carry and transfer multiple drug resistance gene cassettes. Principal role is to provide a convenient insertion site for antibiotic resistance genes. Resistance determinants present on some transposons and plasmids may be the result of integron insertion, facilitating their mobility. These are mobilized predominantly through horizontal gene transfer. Gene cassette contains a single drug resistance gene and a recombination site. After insertion of the gene cassette into an integron with the help of integrase, the recombination site remains available for insertion of a further gene cassette, enabling the buildup of an array of several cassettes within the integron. They move as a unit from one piece of DNA to another among bacterial genome. Differs from transposon in that integrons possess a site-specific recombination system and do not randomly excise or insert into DNA region. The cassettes can be shuffled in their position by excision and reintegration. Through this shuffling process, an integron containing several antibiotic resistance genes can, through trial and error, move the resistance gene that best combats the current antimicrobial challenge to a position where it will be more highly expressed. Classification of integrons based on differences in the gene structure of integrases is class 1 integron, most common type in pathogenic microorganisms, class 2 integrons, class 3 integrons, and class 4 integrons. Class 4 integrons are also known as super integrons. Several classes of recognizably distinct integrons have been found in transposons as well as in plasmids which do not carry functional transposons. They therefore represent a significant additional mechanism for the evolution of bacterial plasmid and the spread of antibiotic resistance. Spread of the tetracycline resistance transposons observed among Neisseria gonorrhoeae, Mycoplasma hominis, and Ureaplasma urealyticum. The recent spread of carbapenem resistance from metallo-beta-lactamases among the Enterobacteriaceae in some European hospitals was facilitated by transposition of resistance genes on integrons. High-level vancomycin resistance in Enterococci is mediated by a composite transposon. Following is the table showing the properties of some well-known prokaryotic transposons. Bacteria can take up the DNA from their surroundings through the process called bacterial transformation. Uptake of naked DNA is active and receptor-mediated process between the competent bacterial cells. Once inside the bacteria, naked DNA will only survive and be passed on to progeny if it is incorporated into the bacterial chromosome. Homologous recombination occurs in all organisms and takes place between two DNA molecules with similar sequences. DNA aligns so that the base pair of homologous sequence match complementarily. Site of exchange can occur anywhere in this region, and cleavage of the DNA molecule and rejoin is precise. No nucleotides are lost or gained. These are the example of gene transfer by bacterial transformation in the environment. What is NDM1? NDM1 stands for New Delhi Metallo-beta-lactamase. Bacteria that produce NDM1 are resistant to all commonly used beta-lactam antibiotics, including carbapenems. To date, some strains of E. coli and Klebsiella pneumoniae are known carriers of the gene. But why everyone concerned? There are currently no new drugs in the research pipelines that aim to stop NDM1. Resistant gene can be transmitted from one strain of bacteria to the another through horizontal gene transfer. Thank you for watching. Do not forget to subscribe my channel and hit the notification bell to remain updated with more such videos.